So shall we start then?
Yes. Okay.
Good afternoon and a very warm welcome to one and all. I am Ms. Zuili Jagtap, Assistant Professor, Pharmaceutical Chemistry Department. Feel privileged to welcome you all on behalf of DPU Pharmacy, Pimpri, Pune, for third International Faculty Development Program on Emerging Trends in Phytopharmaceutical Research. Today's is the seventh session on the topic Neuro-Immunological Disorders, in vivo and in vitro approaches contributing to translational research. For that, we have with us Dr. Manoj Upadhye from Aston University, Birmingham, UK. Welcome, sir. Before starting with the session, I may now request Dr. Ritesh Bhole, sir, Associate Professor, Department of Pharmaceutical Chemistry, DPU Pharmacy, to introduce our speaker for the day. So please.
Thank you, Julie. Very good afternoon to all. It's given me immense pleasure to introduce the speaker of today's session, Dr. Manoj Upadhye. Dr. Manoj is academically proficient with MPharm and PhD from RTM Nagpur University, Nagpur. He is having perfect blend of academic and research experience. He was an assistant professor at Srimati Kishoritai Bhoyer College of Pharmacy, New Kamptee, Maharashtra, India from 2013 to 2015. Later, he joined as a senior executive scientist at Sun Pharma Advanced Research Center, Vadodara, Gujarat for the period of 2 years. Dr. Manoj also served as a postdoctoral research fellow at prestigious Indian Institute of Science, Education and Research, Department of Biology, Pune, India from 2017 to 2019. Currently, Dr. Manoj is a postdoctoral research fellow at School of Health and Life Sciences, Aston University, United Kingdom. Dr. Manoj has published many research and review articles in national and international journal of repute. He is also a recipient of many awards and accolades, such as Overall Best Presentation Award at Early Career Physician and Investigator Conference, United Kingdom, and Best Poster Presentation Award at Encephalitis Conference organized by the Royal College of Physicians, London, United Kingdom in the year 2022, to name a few. Dr. Manoj also delivered many sessions as invited speakers in numerous seminars and conferences, such as Society for Endocrinology, the Annual Conference of Indian Pharmacological Society, etc. He is a life member of Indian Pharmaceutical Society, Indian Pharmacy Graduate Association, Society for Neurochemistry India, and also having yearly membership for Society of Neuroscience. With this very brief introduction of Dr. Manoj, I would like to request him to start with this session on neuro-immunological disorders, in vivo and in vitro approaches contributing to the translation research. Dr. Manoj, please, over to you.
Well, first of all, very warm good afternoon to all you guys, and thank you so much, Dr. Ritesh, for such a nice introduction. And thank you so much for organizing this event and calling me out as an invite. And like, it's really a pleasure to serve this platform. And I would try my best, like, at the end of this session, whatever I deliver might be a clear take-home message as a part of a research career for all this, how would I say, the PIs and the PhDs and the undergrad researchers. And hopefully they might implement these techniques in their lab, and many more to go. Well, so I'll just share my screen and let's start with this.
Yeah, please. Sorry.
Is it visible?
Yes, visible.
So the first slide of PowerPoint.
I think only screen is being shared. PowerPoint is not shared up till.
Just a moment. Is it there now? Or not yet.
whole screen I think you shared, na?
Hmm.
Okay. So now,
I don't know why it's not taking up. Just give me a second.
yeah, please.
I don't know, somehow it's not picking up my PowerPoint here, but I'll just try to go through this. Is it visible
Yeah,
now?
yes.
Perfect.
No,
That
nothing.
makes sense. Okay. So welcome you all for this, like I would say, a highly scientific event. And thank you so much for the organizer for arranging the staff development program. And I would try to take this journey over on the research with the translational aspects, where I would like to discuss about some model developments into the neuroscience research, and hopefully this might be helpful to you in developing and growing your own skills into your lab. And on the last segments of the slides, we will also touch on how you can incorporate this research into your phytopharmaceutical research, and how would you implement with all the phytoconstituents, like emerging phytoconstituents, into this research, and what will be the best outputs. But at least I would say implementing this small translational techniques to the basic research that we do into our daily life in the lab might give you the most fruitful aspects, and they will literally help you to at least get your papers into a, I would say, or rather it would increase the impact of your paper, I mean, really by say 4 or 5 impact. So that's, yeah. And I would also add, like being at Aston here in Birmingham. So while I'm associated with the Birmingham Children's Hospital, where we just look at the patients, and rather I would say a small kids with these disease conditions, that also makes me feel really challenging about this condition, like how it has been developed, and like the way right from the childhood these kids are growing with this neurodevelopmental disorders, and then they are just continuing with, or rather they are learning to survive with it for the entire life. So let's go into it. So what we do here is, like, my simple intention of being a researcher is, I want to take some molecules right from the bed to the bench, and from bench to the bedside. This means, like, whatever the drugs they are available into the market, I would like to take those drugs as an opportunistic element for myself to get them back into the lab, and then screen them out for their, like, all the efficacies, and maybe if they can be combined with some other best molecules, and also been thrown back into the market. Because when we work with the patients, we only have that unique subject in front of us. And like, since there are a lot many clinical ethics and lot many paperworks to go, we can't do much more experiments on the clinics. But the preclinical work gives us an liberty to develop many n numbers with the same conditions. So my lab basically deal with this first part, and like, where we establish the autoimmune epilepsy rodent models. We will go through it, like how we do that. And also we do a lot of in vivo and in vitro techniques to prove whatever research or whatever hypothesis we have incorporated into the work. And after we have successfully developed the pair of combinations which we suggest to the physicians, we get back to the hospitals with the perfect combinations. And this is how it helps the physicians also to think about the best combination treatments and how they can lead further with the patients. So coming on to just developing this model, which is like in our case, what we get is like the CSF samples from these kids from the hospitals, and with our some collaborators in Germany and USA, we get the purified and the isotypes of these antibodies, because of which the encephalitis like condition or the seizure or the epilepsy like conditions are being exaggerated into these kids. So it's not only about the seizures or epilepsy. I would say it's all about the neurodevelopmental disorders. It can be anything. It can be Parkinson. It can be, you can say Huntington's. It can be ataxia. It can be autism. It can be seizure. It can be like and neurodevelopmental disorders which we can relate to the kids. So the best part is when we get the antibodies, we have to think about how we will develop these animal models, because the very first thing is like when you are targeting the central nervous system, the main aim is this component should reach the brain, and crossing the blood brain barrier is always being a difficult and the challenging thing in this particular type of research. So what we do, we have a two developmental models, like first direct ICV injections of this antibodies into the ventricles, and like after 48 hours we just screen their brain, which we call as an acute model. The second one that we have is a long-term treatment of these antibodies for, like you say, 21 days, 14 days, or 7 days, depending upon what animal model we want to develop and how severe or exaggerated conditions we are intending to see. For this, the very simple thing that we do is, instead of injecting them daily, which most of the time we keep on doing, we prime the pumps. Like we use the osmotic pumps. And the best advantage of this osmotic pumps is that you just implant them and it keeps on delivering the drug 24/7 at a fixed rate. So just implant and then forget about it. And these pumps are really easy to like prime. So we call them Alzet pumps. Basically that Alzet is the company which comes with like a flow moderator and some connecting tubes, and then you just fix everything with the cannula. This cannula rather gets seated onto the skull, and the antibody is directly infused into the brain for whatever time you want. Only the thing that you have to take care is, while you implant these pumps into your say rats or mouse, whatever subject you are dealing with, you have to be really careful with your wounds. There shouldn't be any pus, or like there shouldn't be any wound left unattended. So if you see any scratches, just take care of the wound. Otherwise that will be an unnecessary obstacle or unnecessary hurdle, or I would say an unnecessary error into your research. So it's really easy. Just like shave the back, give a small cut, very light cut, because these pumps are just merely at the size of the capsules that we even swallow as a medicine. So just implant these pumps into them and suture the wound back. The tube, it just comes out through the developed, I would say, portion under the subcut, and the cannula has been implanted on the skull for continuous administration. Once you achieve this model, the second thing that we do, since we deal with lot many brain waves, and I have more interest in recording the EEGs, so I'm trying to mimic the conditions in the lab as the doctors do into their clinic. So whenever such patients visit the clinic, the best part, what the clinicians do is like they will just put one helmet like things on their head and then keep on reading the EEG wave for the long time. So that's the main, I would say, the database that gives an idea like what kind of brain waves the patient is giving and how to study them, at what duration of a particular day the patient got those seizure episodes, how long the episodes were, what were the intensity, what were the frequency of the brain waves, and how to deal with them. Okay, so what we do here is like once you have implanted like the rat with the osmotic pumps, the best thing is to just put this one of the transmitter depending upon the size of your subject. So they really vary from like 1 centimeter dimensions until like as big as you want, like 2-3 centimeters, and then they comes with the recording wires. One of the wire has an electrode point, or rather I would say a D point, which gets attached to the electrodes, and those electrodes can be targeted to a specific brain regions from wherever you want to read the EEG waves. Say in my case, I would target the electrodes to the hippocampus, because we are also looking for the neurodevelopmental disorders, like sorry, neurocognitive disorders, okay, in the developmental phase of these rodents. And the rodents might be, since we deal with kids in the hospitals, we also keep these rodents like at very low age, maybe day 20, day 21, which is really a developing phase for them. And that's the perfect age to develop any, I would say juvenile model. Coming on to the recording of this system, these transmitters emits the waves which are then captured by the antennas and the recording system, and this recording system throws out the signal into the computer in the form of brain waves, and these brain waves can be analyzed like by using software which are freely available by the companies. So this is really an easy thing. And I mean, once you get the receptor antenna and the octave focal box, you don't even have to pay for the softwares. The softwares are really available in the market. So this is how it looks like. So I would say, like, there are your cages. These are the antennas which receives the signal. And the best part of this technique is the animals, they are freely moving in the cage. Like you won't see any wire or anything, because you are using the wireless telemetry procedures to record them. So there's no pressure of the wire, or like the animals biting out the wire and then just tearing apart them, and then we lose the entire subject out of research. So no such headaches in this particular techniques. And at a time you can build as many cages as you want, depending upon the octal receiver that you want to get from this company. So based on this like antennas which we place into the I would say Faraday cages. So why Faraday cages? Because we are dealing with the EEG waves from the brain, and we don't want unnecessary radio frequency measures. Say suppose if I'm having my mobile in my pocket and then I'm trying to handle them, the 50 hertz frequency waves which are being emitted by this electronic device will also be captured by this system. So somehow what we will think, like, this might be a signal, or this might be a seizure, or because they are really high frequency, like 50 hertz you can think of. And that's how the error occurs. So most of the time you have to be careful, and that's why you have the Faraday cages, which really obstructs the surrounding radio frequency devices noise getting involved into your brain waves. Then from this antenna, the data is being received by the LWDAC drivers. That's the name of, how would I say, the software and the drivers both. And through the cable system, they are being recorded into the software signals, and that's how you analyze them. Just take them from there, read the seizure episodes those your animals are having, what is the duration of this seizure episode, how long they were staying, and what is the frequency of these waves. So when you talk about the frequency, it's all about the measure, like the peaks of the waves. Like you can see in this particular example, I mean, we'll come to the specific examples as well in this presentation later. So when you look at the controls, it's really a stable line without any, I would say, a lot noise or something, and then no such big peaks and all. But when you look at the epileptic guy, you can really see a pattern where there are bursts of all these episodes, and you can easily make it out whether your animal model has worked or not. Instead of waiting for the end points, which sometimes is like 45 days or 50 days or 60 days in most of this I would say neurodegenerative cases, and at the end when we don't see anything, we are disappointed with whatever time we have invested, whatever work we have done, whatever inputs we have given, or whatever things we have purchased, which are really costly. So the best part of this model is like there are everyday analysis that you get, and by seeing them very quickly, you can make it out whether your model is working or not, or you might to increase the dose of antibody or decrease the dose of antibody, or give more drug, I mean give a valid combination, or you have to change the drug over the time. So yeah, this is the perfect example of, I would say, in vivo recording. And this slide now represents the in vitro approach that we do to study in more detail, like how these brain samples would look like, or why this particular brain, like why this particular animal started shooting up. So in case of epilepsy, studying cortical brain regions or the hippocampal brain regions into more detail, and the neuronal connections into more detail, helps us a lot. So we have a two approach system here, where we first do the single neuronal study, where with the help of like a small glass pipette, which we call electrode, we try to enter the arena of the cell. And once we do that, the best thing that we can know is like how big this cell is speaking to us. I would just say speaking, because that's how the internal environment of the cell is. Is it overshooting? Is it undershooting? Or, like what is making it to communicate more with its surrounding neurons? So that's what about epilepsy is, because in epilepsy, the neurons start talking to each other a lot as in a lot. And in as compared to other conditions, and that's why when the brain gets confused with all such informations which they can't gather at a go, or where they can't differentiate in between the two informations which are just shooting at them, it starts behaving weird. And that's how, if it is a motor cortex or the brain region which is controlling the movements and movements of the limbs and other body parts, it start giving the tremors, that's we call fits. So basically that's an underlying mechanism, because the neurons start shooting very badly. And this is how we can understand why they are shooting out, whether it is NMDA, whether it is GABA, whether it is serotonin, or any other brain basic neurotransmitter which has been involved into it. Or there can be most of the ion channels like sodium, calcium, potassium, which are involved into it. So once you get onto the patch, you try to understand the internal environment of the cell, you can manipulate the internal environment, and that's the main basic thing of this drug development thing. So here you can see, if it would be a control brain and then you could get into the internal environment of the cell, it will still behave basic. And to be honest, this replicates to the one in vivo EEG recordings. Okay. So when it is control, it is control. Either it's an in vivo model or it's an in vitro model. But in the epileptic condition, the, I would say, neuron, they are overshooting, and that's why you get a high hertz, or rather the high frequency or high amplitude peaks, which you really want to study and then know. Like, and the best part of this is, like, if you start treating them say in the dishes, like in vitro, all these epileptic spikes that you can see here, like in the upper portion, they will go down. And that's how you can also confirm whether your treatments are working here or not. Okay. And the second part of this slide, where I have written local field potential, this basically we do to understand the communication of one brain region to the another. And that's what the main, I would say, stand for this research is, because as I mentioned earlier, in epileptic condition, one brain region starts communicating to the other brain regions in a broad way and like uninterrupted way. So the second region, which has been receiving or trying to perceive all the informations being thrown at him, instead of, like, getting the segregated informations and then just splitting them out, trying to understand what's going on, and then giving us an time to react to that. That doesn't happen in the epilepsy, and that's why it starts giving tremor. So when you look at the control trace here, it has the informations which has been passed from one region to the another region. So in this condition, we placed electrode at the CA1 region in the brain, and we were just trying to look for some memory coordinations from this region to say other like dentate gyrus or CA3, or maybe other temporal lobes or the cortical regions. So what we saw was like control was still good, but then in the epileptic, you can see subject, it's a lot of shooting in that. And how this help us is like maybe we, as I mentioned, we mimic the patient's physiology here, and then we can always get onto the treatment regimens. You can mix more than 2 or 3 drugs into the bath, and then you can always study, you can always manipulate the doses, and this will really help us understand the internal mechanisms, not only seizure, because I am right now actively working with this patient. So I have tried to incorporate this as an, I would say, a prototype model to make the things clear to you, like how we can reach it out. But I think this might be the same conditions with any neurodevelopmental disorders. And you can also study the target receptors or ion channels and manipulating some internal genes, or maybe, like getting onto the knockouts or any other strains, might give you a really good idea whether that gene is involved into, I would say, this behavior or not. Also getting some, like basic idea about understanding the compatibility of the medicines that we give to the humans, and why sometimes like some humans are drug resistant and they do not respond to the treatments. We can also develop the same models here. And this prototype of research, like if you would like to go for your future drug development into, say if you are approaching the US FDA for getting a new drug applications and filing some, or throwing some molecules into the clinics, this part of in research, or this, I would say, firm evidences of the research really help us good. So as mentioned earlier, when I was working for the Parkinson's research with Sun Pharma, we had this, I would say, some important bits of informations which helped us getting one of the molecules straight away through the US FDA, and then it's in the clinic now. Well, coming into more detail of these aspects, I would like to share this using one of the antibodies that we recently did like a year ago. So where this idea came from was basically this polar bear, the one that you are seeing on the right hand side, rotating. So this guy, basically named as Newt, was a bear in the zoo in Berlin, which was really like, put like given away by his mother. And she was no more taking care of this guy because he was behaving abnormally. And one of the scientists, like called as Bruce, when like they visited the zoo and then they saw this particular behavior, like this is not something normal. And you can see the rotations, and at one stage you will just see that. So this one, it's a typical measure of seizures that you can see, and the guy falls down. So this is what I want to say. So the scientist proves, when they visited the zoo, his group, they got an consent for this bear, and then they extracted the CSF from the samples, CSF samples from this guy, to understand what's the main cause of this behavior. And they came to know, like, this is a mere condition of autoimmune disease, where the body starts developing the antibody against one of your own, like neurotransmitters, or like neuropeptides or neuroamines, or in whatever way it is. But body doesn't make a difference, or like it thinks the internal or the biological amines are itself an foreign invaders, and then it starts developing an antibody. So as a result, the NMDA receptors get internalized, like they are no more available for functioning properly, and that's how it leads to hypofunctions. And this is the main reason why it keeps on shooting the NMDA. Now, as we know that NMDA are involved in the iPSCs, no, sorry, they are the stimulative factors. The inhibitory ones are the GABA. So the GABA goes down and the NMDA goes really badly up. And because of that reason, the behavior was such weird of this animal. Eventually, like, it died, say, after a year ago, I guess. No, not a year ago. In 2018, it died. But before that, they could successfully work on whatever the informations they could get with all the consents and everything from, and all the ethical measures they could do. But this helped us getting into this new world of the autoimmune disorders or the antibody disorders. So this is the similar model which has been developed in our lab, in fact, developed by me. So as I earlier mentioned, like, you can have the two measures or the two approaches here. Either you inject directly into the ICV, and then you can always have, like, this is a short term and the acute protocol where you don't need any kind of transmitters or osmotic pumps. Like, I understand, like, the financial budgets are really like most of the important things in the research. So if we don't have that, you can always go for the short and sweet experiments, which are the ICV injections, and they still will develop the same conditions because you are injecting antibodies into them. And the other part is, as I said, like a long-term experiments to see how it goes on, and then in these conditions, how the treatments will taken care of, and then what are the best combinations that you can give. And in this particular guy, what we did was, like, we recorded the behavior. We also did some neurocognitive tests. We also did the post Caesar's behavioral battery tests. And after that, we got the brains out, and where we did some in vitro experiments. So just from one animal, you get a lots and lots of data, in vivo, in vitro, everything. So this is what the observations and the the behavioral observations and the in vitro observations were for us. So as I mentioned, the control with the NMDA antibodies, and in the normal, like, in vivo EEGs. And this is the local field potential experiment, like, where you can see control, there was nothing. So control, as in, this was not a naive control or a normal control. But when we ordered these antibodies from the collaborators, we also ordered their cohort, like, the specific control cohorts for them. So they sent the controls but without the antibodies. So they also had the CSF samples but from the healthy volunteers or healthy patients, and they were also derived isotypes. So it wasn't like we just injected some, like, PBS or CSF, and then we were trying to prove the control, but they were also derived from the patients, from the healthy volunteers, and then the antibodies from the patients. So the second part, where you can see with this antibody how drastically it changes, like the lots and lots of bursts of these events. And that's why the rat you saw in the earlier video was jumping very badly. And the simple thing what you have to do with some, I would say, software, not even some softwares, like, you can always use ImageJ, as in the Fizzy, and how would I say, the spike count softwares, which are freely available, and which will give you the measures of the spikes. And if you count the bursts in like 15 minutes, that will give you a clear idea about whether you are having your antibody model or whatever model you are intending to, it's working or not. So here the simplest example of this is the spike counts, where you can't see much in the controls happening, but at the same time in the antibody treated conditions it has been shooted up. And in addition to this, I would also like to say, like, when we discuss about this model development at various platforms, I was being asked several times, like, how do you confirm, because you are injecting antibodies into these guys, And how do you know? Like, because the behavior, whatever, or the changes, whatever you are getting, are because of these antibodies, or they can be an artifacts, they can be anything, whatever you see, or it can be an impact of surgery as well. So the best answer to that is, I would say, the microscopic sections or the microscopy image that you are seeing right now. They are simple sections taken on either the fluorescence microscope or the confocal microscope, if you can get. But they, like, since these antibodies were human derived, we tagged them with, like in the on the slices, we tagged them with the secondary antibodies, which were anti-human and green, of course. So Alexa Fluor 488. And as you see, a major difference here, how much of this NMDA antibody thing is illuminated, totally green, green, green. And the other one, which is the black image, is of control. So that's the control antibody. So this gives a very specific idea about the model, whether it is working or not. So behavior, in vitro, and now you got the microscopic assessments as well. So that really forms out of whatever observations you are getting, and you are quite confident about what you are seeing. So this is from the human samples, like what we saw from one of the patients from the children's hospital, wherein the patients, you can see, like, so in control it was really staining good, all the NMDA receptors, but in the patients they were really internalized. And this is how the journey started with. And then in this patient, so the antibodies derived from these patients we injected into the mice, and then we have this cohort here. Like, and I'm so glad to say, like, it every time works for us in whatever species it is. So the strains, like the rats, mouse, and whatever strain you want to. So here we have used the blacks, we have used the black strain, and the rats were like a normal V star rats. So they always work fine with us. And so why we are doing all these things? So just to get into some medicines, or what are we trying to do here? We are trying to develop some new molecules as a part of our research, how to say, journey, or we are trying to evaluate some already available drugs into the market, or we are trying to fix out the best combinations for the patients who are struggling out there on the beds. And like, whatever way, whether it is an synthetic drug, it's a natural drug, or from wheresoever, whatever the source we derive it from, the main aim is to get rid of this condition. I know it's really a wrong phrase to get rid of, but at least to minimize the suffering of that particular human or the patient who is right now on the bed. So that's what the main aim of this research is, and that's how I call it as a translational journey to the research. How we can rescue these things? So we have simply our receptors to which the antibodies were developed. Because of these antibodies, the receptors were internalized, and then there were no receptors available to accept whatever treatments we are giving, or whatever, like, challenges we try to give. So the best thing is, like, you get rid of antibodies from that patient, which is really impossible. You give them such substances which will reduce their conditions, which is okay. We can do that. The third thing is that we can also overexpress our receptors by giving some or the other, like, medicines or the drug, which will really help. Say, suppose we were needing, like, we were having 100 receptors as in previously, which were internalized. So instead of having 100 receptors, why can't we give such drugs or treatments which will, like, generate or help in getting the more receptors onto that field, say 200, 300, and then we will never be less on the receptors, and in that way we can also keep a hold on this particular diseases. So there are certain, like, I would say, neurosteroidal molecules which are available in the market. Say one of them is pregnenolone and alloprex and all those things. So they really help good in getting. And one of the best examples that I mean, I can say is, if you just uh, go with this immunolabeling things under the microscope, you will see like in the patients how bad these receptors are, and after these treatments, are they really expressed or overexpressed, or they are in the good shape. So once they are like available there, you can also suggest these treatments to the patients. And like, I'm so lucky to tell you that the doctors here, they accept willingly all those treatments. And as I said about the pregnenolone sulfate, so you can see here like all these bursts in how would I say, in the local field for the rats that we developed, they were just taken down with this neurosteroidal molecule. And it helps really well to get like into this model. So again, in the spike count and the inter-event interval analysis, you can see like how well it is, like working, and I mean how promising this molecule is. So that is that was the one thing. And also, whatever drug you take or you want to inject into like the subject, we have to always keep one thing in mind. Finally, we want to take this molecule to the patients. We can't give them directly into the brain. We can't like every time how to say, for have some forceful injections or something to the patients. The best part is like, as nowadays they got really a good subcutaneous pouches, and the best drug delivery systems out there. We just have to make sure that whatever drug you are intent to give them, it crosses the blood brain barrier and it reaches the brain where we exactly needed that. Okay, so for that you can always take out like some additional experiments, and you can always do some mass spec to understand the brain levels of this drug in controls as well as like non-treated ones, and then the drug treated. Let me tell you one thing, like when we did this experiment, so we saw really good amount of pregas, and then we also had cholesterol as an like control molecule to check whether it has been affected with these injections or not, which it didn't. And it's not always like you have to do everything as a researcher. The best part of being into the research is you can collaborate with some other laboratories. The one who is expert, say suppose this data wasn't generated by me, the best part what I did was I gave brains to them. I requested them like, can we please work together to get these data out, and then this will benefit us both, like both the labs. So you always have to figure out the best collaborators to reach out, like in what best way I can carry out my this research. Is it possible to work with you? Is it possible to get onto this data with you? Like this will benefit us, and then you can always throw a joint grants together further based on the pilot data or getting on whatever data you want. It's it's about collaborating. You can't always run and then develop your facility very highly, or you can get everything into it. You can't be doing everything on your own. Coming to the phytochemical constituents, what recently it is trending out, like a few of them we have also did in our lab now, I would say. So like lot many drugs are available into the market, not even market, like lot even like the vendors are been selling the extracts, like whatever way you want. If you want leaf abstracts or extracts, or you want the root extracts or whatever. So just the thing is like you have to reach out to a proper vendor to see what you want. So or maybe um, there are some flavonoids, alkaloids, glycosides, all these things also, the extracted form, which are available already, but they cost a lot. But I mean, if you can extract them into your laboratory, it's the best part. But if you can't, there's always some resources available to reach them and get them from them. So what we did right now, and why we were doing this, is because what we understood is like there were some flavonoids like phenylpropanoids and terpenoids which really were helpful in this epileptic like conditions. And Also, they were targeting the GABA A receptors and the voltage gated ion channels, basically the calcium ion channels, which might be of really good interest to us. And there are like also many derivatives outside there, which also will give you, say, a neuroprotective effects. Why neuroprotection we need? Because most of the time when we are dealing with this neurodegenerative diseases, they go for the neuroinflammation. And one of the strategy to get how to say these conditions elevated is decreasing the inflammation as well. So if you try to hold on the inflammation, it might also give you that opportunity to overcome the disease very nicely. Recently from our lab, like we tried with the cannabidiol, and like of course it's an cannabis and the euphoric constituent, like non-euphoric constituent, but it's from the cannabis. This is one of the best phytochemical substrate nowadays, which is in a trend here for dealing with the epileptic like conditions. And I think last year, or yeah, last year, so there was a group from the Rosenberg which has developed a good research on the cannabidiol in the seizures, where they have done the multiple models. So they have done the pilocarpine models, then the strychnine model. Sorry, I don't know what was that. So the pilocarpine models, and then the strychnine models, and like lot other things, even the mechanical seizures also they have done, and then they have reported like how this cannabidiol is really effective in mechanistic way, how it get control over the calcium channels, how good it is like controlling the brain EEGs and everything. So that was the best research which they have done. Why mechanisms which we want to deal might be out of one of this only. Either it can be some neurotransmitters, some receptors, some synapses and channels, inflammatory cytokines, maybe glial cells. The most important one right now for the neurodegenerative disorders is the oxidative stress. I know most of you might agree to it, because in India also right now the oxidative stress has taken a quite big, like area of research, and some mitochondrial dysfunctions and the genetic factors. That's what I mean to say when I was talking about some knockout mice and then the genetically manipulated mice. So if you can also collaborate with some companies, or the industries, or from the animal suppliers who can breed some genetically knockout strains for you, that can be also really helpful to grow your field into the research. So that's it, yeah, from me. And I would like to conclude on this particular topic into the translational world, and I'm open to any questions and happy to answer. Also, we are open to collaborate if someone wants to reach out to us, and I'm always ready and keen to help in developing some of the animal models, not only in epilepsy, but since my area of expertise is neurodegenerative disorders, feel free to reach me out. Like if you need any help, or like any advices in developing such models. Well, I would say thank you on that. Over to you, Dr. Ritesh.
Yes, yes. So if anybody want to ask some questions, Dr. Manoj is happy to answer it.
Dr. Ritesh, am I audible?
Yes, yes.
Is there any kind of computer-aided approach that can be applicable for the particle states in terms of the disease which has been covered by the Dr. Manoj?
Yeah, Dr. Mavir, you want to ask some questions?
Yes.
Yeah,
Yes.
please.
Is there any kind of computer-aided drug discovery approach which can be applicable to the phytoconstituents for the disease patterning which has been covered by the Dr. Manoj?
Manoj, you are muted, Dr. Manoj.
Yeah, actually, I couldn't hear. So there are like 2-3 voices which are coming out. Like,
Dr. Mavir, can you ask again?
sorry for that. Like, yeah, Dr. Mahavir, can you just repeat that question, please?
I will type in a chat box. I think there is some disturbance.
Yes, please. That will be a good option.
Yeah.
So till then, can we take the other question?
Yes.
So the question is, which are the potential targets for drug discovery?
So depends upon like what type of disease you are dealing, and I would say if you are thinking about the drug discoveries, you can basically go for, say, in like neurodevelopmental conditions. It's always been either like the genetic sites or the receptoral sites or the ion channels, which are mostly involved. So if you have some neurodevelopmental disorders or the neurodegenerative disorders, the very first thing to think is like, are there any ion channels involved in that? If there are some ion channels, then just go for the screening of them. The second thing I would say are the receptoral sites. There are really, like, when we talk about a specific disease, there are really few receptors that we can see. No doubt, the underlying mechanisms of these receptors might be really different, might be some kinases or some, like other profiles which are involved as in down regulatory mechanisms to that. But that's the secondary story. So whenever you think about the drug discovery or, like specific targets, the very first approach I would say to go for the ion channels and then the receptoral sites. So just to start with, that's the best thing if I answer that question.
Yes, thank you, sir. So the question by Mahavir sir is, what's
Yes.
computer-aided approach we can correlate study of phytochemicals with CNS diseases discussed today?
Okay, so again, I would say what type of neurobehavioral disorder you are trying to target. So if it is epilepsy and then we are dealing with the brain waves, then there are really some, like, open source instrument softwares available in the market which are free. So you just have to go explore them because you are dealing with the brain waves. But if you are dealing with some other like part of the neurodegenerative diseases, say Parkinson, or where you have to score the subjects, maybe how would I say, manually or through some computer aided graphics or some softwares. So there are basically some softwares which are available. To name with, I think there is. I'm just losing the name, but for Parkinson's disease, when I was working actively with Sun Pharma, we got a really nice software. So you have all the behavioral symptoms like rearing, tremors, not even tremors, but yeah, I would say tremors because it was the shaky behavior of the animal. So then rearing competitiveness, tremors, its ambulations, then the locomotory parameters, also some neurocognitive functions. So all these things, and then they're basically, how to say, the limb placement tests. So for all these things, there were some computer aided, like it was one software, but I don't remember the name. It's a long back now for me, and being into Parkinson. So yeah, there are various things, but definitely I'm happy to help you out getting out the specific informations if you need. And then I don't know how good it is to say to add MATLAB into this analysis. So, whenever we are taking some scores or something into considerations, and then we are getting through the MATLAB scripts, they are really good and the genuine one, because when you submit the scripts to the publications or to like whatever resources you are claiming to get onto this work, they are genuinely accepted and they are really happy to like validate them for you. So if I answered that question, Dr. Mavi, for you.
Can I interact with Dr. Manoj? Am
Yeah.
I audible?
Yes,
sir.
yes, you are.
Yes, sir.
Dr. Adipalli, it's so nice to listen to your work with patch clamp techniques, which I used to do when I was in, postdoc studies. As
Oh,
you
wow, that's lovely.
rightly mentioned your title, it's more of neuro-immunological disorders.
Yes.
And we can even come down with myelitis, multiple sclerosis, today even optica, number of such disorders are there. In fact, they are
Yes.
rare as well. What I enjoyed in you have shown the preclinical study more today, how to diagnose clinically, what are the various recent techniques, number one. Number two, my question, when we are looking as a chemist and a pharmacologist, how to repurpose a drug mainly working towards ataxia, telangiectasia, because the reason, Manoj, I had wonderful one project as a expert supervisor. Recently, I cleared ICMR. It is mainly using carnitine and betamethasone for ataxia, repurposing. So third one, a part of that, human stem cell technology also is taking place in neuro-immunological disorder. So I want Manoj to throw light on this.
Okay. So coming on one by one, like talking about the stem cell things.
Yeah.
So the best and the most important part of the stem cell things is the iPSCs, like induced pluripotent stem cells, which basically in this type of my how would I say area right now where I'm involved in, where we have the kids having this particular diseases and disorders because of the antibodies which are generated. And if we target some of the stem cells, or if we start like refurbishing some of the pluripotent stem cells into them, which can take care with the binding sites, because most of the antibodies, they have the peripheral receptors as their binding sites. So if we can have this stem cells adjuvanted with the peripheral receptors for these antibodies, the antibodies can be diverted to these stem cells, which are like hypothetically organized for them or like mimicked for them, instead of they binding onto the actual receptors.
Yeah.
So that's the another thing, because when we have these antibodies into our how to say on our table, not into but on our table in front of us as an isotype in front of us, we can always go for their genetic sequences, and then we can always go for their how to say the molecular dockings. Once you have the docks available, you can always refurbish your stem cells, and then you can inject directly them into the like ventricles or the brain. So if that can be a really useful thing to do, because one of our collaborators in Germany, they are onto these experiments, and every now and then they keep on making some or the other epitopes for these antibodies to have them
Because that is
binded.
option nowadays, nowadays that is another option is there. Actually, one more, how do you bring out some diagnostic part in a patient suffering from multiple sclerosis, a kind of neuro-immunological, because it is a challenge for most of the clinicians to come out with some particular test.
Yes. So for that, we have an entire battery of forms available with us. When we have a particular patient case in front of us, what we do is like the very first thing, we ask them to screen them out. And we have many behavioral, like, I mean, not to exaggerate, but it's a totally 8-page forms for us. And then since they are the kids, we need their parents with them as in part of their consent. And it's a seriously how would I say exhaustive battery of the questions which we ask them very minutely, each and everything in detail. And once we have them, we also have the consents to get the blood samples from them, the CSF samples from them. So we have a good facility where with the help of all these neurologists, we extract their CSFs, and this CSF partly is being taken care of by our lab, and then partly to extract the antibodies by the collaborators. So on this CSF also, we run a lot of molecular things like Western blots. You can also do RT-PCRs and all these things. So to understand what genes have been differently, involved in these conditions. Also, when we get an extract messages about or the differentiations about all the antibodies from the collaborators, like what are at the peak or what are like going down. So that also helps us to confirm our how to say our like validate our observations as well, and then we get back into the meetings with them.
Good. Lastly, just one more kind of interest. When you said MRDA, that cannabidiol, I heard earlier, as you know already, there is anandamide, which is a endogenous probe for cannabinoid receptors. And this anandamide can bring much more meaningful interaction through NMDA and cannabis. And this is one area of good research. When you were talking very nicely on that patch clamp, and naturally ligand-gated ion channels, if your electrophysiology is such a wonderful, authentic data, one can come out. It's a beautiful thing. So anandamide can be one of the useful probe. That's what another one more. Today, repurposing is one thing. Could you come out and say, as a chemist, as a pharmaceutical person, can we reinvent some repurposing drugs for such rare disorders?
Definitely we can do that. And that's the another challenge being as a translational scientist. So that's why we call ourselves as a translational scientist instead of basic scientist.
Thanks, Manoj. I will talk to my department HOD and we will try to see how best we should collaborate, take forward, and this kind of research is very always important to bring basic structure and add on more value to the scientific arena.
Yes, definitely. Thank you so much.
Thank you, sir. Our next question is, are there any phytochemical based therapy currently available for CNS disorders?
There are a lot. There are seriously a lot. So even if you just Google out like the phytochemical constituents in dementia or, cognitive functions, you will find many. That's what I shared in my slide. Like those two slides, if you see clearly, those two tables were full of phytochemical constituents, different drugs, what part of that plant, like leaves, roots, extract, whatever it's being used, and where it is specifically targeting, and then what they are checking at, whether neuroinflammations, whether cognitions, or like whatever part. So right from the behavioral until getting onto these deep details into the brain or deep details into any body organs, not only about the brain, but any body organs. So it can be really helpful.
Next question is, are there any funding agencies to support neurodegenerative disease research?
In India, yes. You can also, you can always go for like Department of Science and Technology. You can also go for ICMR. You can also go for some MRCs and like medical research grants. And if you want to really like think about collaborating with some abroad agencies, and then you are always happy to throw some of the grant, like some of the work into the European grants, but you have to sit and look at it. And like maybe some, like states-based grants or like this United Kingdom-based grants. So, but they are really open, like if your work's really valid, and then sometimes they really give you the fellowship for 6 months or a year to come over the lab and then work there and just authenticate the data, and you can always publish it with them and then go back. So that's how the things work here.
And our last question is, challenges that you have faced to address capacity to cross blood-brain barrier.
Challenges. There are many. Like, so basically, when we are intending about the patients, we always think about giving them anything orally or either injecting them. So the very first thing that when I started like my career as a neuroscientist back in India, we had those challenges getting the molecules reached into the brain, because not everything is like easily crossing the blood-brain barrier. So that's why we started most of like my research was basically on neuropeptides, where we know like peptidergic analogs are really big. They will never cross the blood-brain barriers. But somehow, if you get the synthetically available peptides, which are accepted by the lipoidal membrane of the, like BBB, so that can easily access not the entire, I would say, the dose that you are injected, but at least it allows 15 to 20% of the drug to enter the brain, which is sufficient for us. So to be honest, if you are giving something orally 100 milligrams, I would say the quantity which should reach brain, you won't even expect. Like sometimes 100 micrograms can also work efficiently out of that. So whenever we are doing direct injections into the brain, it's always that's less dose, like 100 or 200 micrograms, not even 1 milligram I'm talking. But when we are taking orally, it's a lot. So that's why we use these approaches, because sometimes these peptides, these antibodies, they are really expensive. So directly injecting them into the brain validates your model. But then when it comes to in real world, you always have to be thinking about orally or like injecting them. So there are always synthetic ways to build those.
We have two more questions. So the first is, can you please suggest models for anti-migraine activity for evaluation of mechanism of action?
Anti-migraine.
Yes, sir.
Okay.
Anti-migraine activity.
I have to think over it, because migraine is something that it needs a parameter, it needs a measure to be readed. And in rodents, yes, but in patients, it's always easy. So it depends, like if we are working with some rodents, or even, the best thing is like if we are heading towards any higher species, like non-rodent species, say macaws or monkeys or chimpanzees or something, it's really easy to deal with them for migraine like behavior. But if apart from behavior, if you want to check something, then always go for the neuroinflammatory markers in the behavior. Neuroinflammatory markers in the brain. At least what are the migraine markers in the brain, or maybe the blood samples, that can definitely give you an idea about, developing a model. But reading migraine, something in rodent, I would say it's difficult, because they can't express how severe headache they are having. There will be always a level of some markers like interleukins or maybe other inflammatory like neuronal substrates.
So the last question, shall we expect rapid onset of action with phytochemicals in neurodegenerative diseases?
It's a bit difficult to have a rapid onset of action. And that's why whatever the nutraceuticals we are thinking of, like the natural products, they are supposed to be taken in a long term. So it's really difficult, but depends also like what extract it is. And yes, one more thing that can be achieved, but depending upon your career, or career as in like the vehicle that you are using. Say if you are taking an ethanolic extract, and then we know like how quickly ethanol can cross the like out of blood, how quickly it can reach to the organs, not only brain but other organs. So yes, I would say sometimes, but depends upon what vehicle. That's why when we get all these kadas and everything into the bottles, you drink those, it has always like 50-60% of ethanol base, and that's really helpful to reach out to the target sites quickly.
That's it, sir. Thank you so much for the very wonderful and advantages of it. He explained about the transmitters implants for EEG recordings. He elaborated on in vivo and in vitro models of epilepsy, like whole cell patch clamp, local field potential, NMDR antibodies model, and how to read the output by spike responses. He also gave some examples of phytoconstituents which are present in epilepsy research and the mechanisms involved in epilepsy. So thank you so much, sir, for such a wonderful session. Kindly accept this certificate as a token of appreciation.
Thank you so much for this opportunity, and like personal thanks to Dr. Ritesh as well for reaching me out for this lovely platform where I can share about the expertise and it was really lovely to have you all together here.
Thank you so much, sir.
Thank you, Dr. Manoj.
Thank you so much, everyone. Wow, that looks really amazing certificate. Thank you for that.
I deem it a great honor and privilege to propose the vote of thanks for the 5th session of 3rd International Faculty Development Program. On behalf of our institute, I would like to extend my heartfelt gratitude to our esteemed speaker, Dr. Manoj. My heartfelt thanks to Honorable Dr. P.D. Patil, sir, Chairman, D.Y. Patil Unitech Society, Honorable Dr. Somnath Patil, sir, Secretary, D.Y. Patil Unitech Society, respected Dr. Sohan Chitlange, sir, CEO, Administration, D.Y. Patil Unitech Society, and Principal, DPU Pharmacy, and respected Dr. Santosh Bhujbal, sir, Vice Principal, DPU Pharmacy, for their leading supervision and inspiration at every point and providing us all the necessary facilities to carry out this event. I would extend my generous thanks to all the delegates for their participation and cooperation. I take this occurrence to thank the entire organizing committee and technical team for their unstinted support in making this program a success. Once again, I thank you all for the attention. Thank you so much. Now I request the participants to please switch on their videos for the group photo. Thank you, everyone. Thank you so much, sir.
Thank you so much. It was a pleasure having you all.